like to welcome everybody to the first day of our symposium, which is focused on importance and challenges of increasing ethnic diversity in human genomics research. On day one, we're going to focus on the importance and value of increasing ethnic diversity in human genomics research. And tomorrow at the same time, between noon and 4 p.m. Eastern Time, we'll be discussing challenges with conducting genomic studies in minority populations. This symposium is co-hosted by the Penn Center for Global Genomics and Health Equity, which is directed by myself, Sarah Tishkoff, and by the Department of Genetics at Penn, for which Dr. Daniel Rader is chair. So I am so excited when I saw the number of participants we have. We have nearly 900 participants from all areas of the globe, maybe a few less in East Asia. I think the time difference is playing a role there, though I'm impressed that we have some people from Australia, Southeast Asia. And I'm so excited to have so many participants from throughout Africa. You know, this has been a tough time with COVID, but I suppose part of the silver lining has been that we've had these video events, virtual events, where we can have such a broad inclusion of people from across the globe. So I'm really excited about that. And in addition, we have speakers who come from throughout the globe. I wanted to start by telling you a bit about what motivated us to set up the symposium. And that has to do with trying to understand risk factors for health disparities. So let me just give you a couple of examples. We know that in the US population, hypertension is more common in people who have African ancestry compared to other ethnic groups or other ancestries. People who have Native American ancestry have higher prevalence of type 2 diabetes compared to other ethnic groups. Now we know that there are social, socio-demographic, economic, and other social determinants of health and things like diet that are strongly influencing these health disparities. But it's thought that there may be genetic risk factors as well. And so one of the goals is to try to distinguish the genetic from the environmental factors that influence health disparities and how they may actually be directly or interacting with each other. And that's something that I hope, will be part of our discussion today. And of course, one of the most striking health disparities right now is what we're seeing for COVID-19. I pulled this down last night from the Center for Disease Control, so it was updated February 18th. And we could see that if we look at the number of cases, it's almost double the number of cases relative to people of European ancestry in American Indians. If we look at the rate of hospitalization, it's threefold or higher in American Indians, people of African ancestry, Hispanic or Latino ancestry, ethnicity. And the mortality levels are strikingly higher, but at least double in these populations relative to people of European ancestry. And undoubtedly, we all know that socio-demographic factors and frankly, systemic racism is playing a major role here, including unequal access to healthcare. One of the things that we don't know is what are the factors that are playing a role in why some people are getting mild symptoms and some people are getting severely ill. It's possible that there could be genetic factors playing a role, and we still don't know if these might differ amongst ethnic groups, but that is not to say that genetics is playing a major role in the prevalence of this disease. It is clearly social determinants that are affecting that. So this is from a perspective we wrote in Cell in 2019 with some colleagues, Giorgio Sarugo and Scott Williams. And when we looked at the Genome-Wide Association study database at that time, we saw that 80% of all the individuals used in those studies were of European ancestry. About 10% were East Asian ancestry, about 2% African ancestry, 1% Hispanic ancestry, and less than 1% everyone else. And we felt that this is really problematic because it impedes our ability to fully understand genetic risk factors for human disease, and it could actually exacerbate health inequalities. Our ability to translate genetic research into clinical practice or public health policy could be incomplete, or even worse, it could be mistaken. And attempts to use estimates of genetic risks, such as the polygenic risk scores, that are often typically derived from European-based studies, don't translate well to other ethnic groups, and they may actually result in inaccurate assessment of risk and lack of appropriate interventions. And I also should mention that it also, when we determine what is a pathogenic variant, often that is determined based on how common a variant is. So if it's very common, it's thought that it's less likely to be pathogenic. But maybe a variant is not common in Europeans, but it's very common in other populations. So that literally could change what we are classifying as pathogenic variants. I want to take a moment to tell you about the Penn Center for Global Genomics and Health Equity, which we just established in this past year. This is Dr. Dorothy Hammond, Assistant Director, and Amina Alamin, Executive Assistant. I want to tell you a bit about the mission of our center, which is to promote health equity through global genomics research, education, outreach, policy, and practice. We aim to facilitate and promote research that's focused on both genetic and environmental risk factors and how those interact to influence health disparities in the U.S. and across the globe through interdisciplinary collaborations, community partnerships, and education. We ultimately want to translate these findings into public policy and clinical practice in order to promote health equity. And we want to promote inclusion of ethnically diverse individuals in genetics research and understanding of ethical, social, and political challenges that arise when we include minority populations or indigenous populations in human genomics research. Another thing we think is really important is the education and outreach component. And as part of that goal, we're going to be hosting workshops in the future to train people in things like bioinformatics skills, and we want to have a global reach. So please check out our website, which is listed here. If you want to be added on the listserv to get email about it, just go to the website and there's a place to do it. And please do tweet about the symposium today. So a few guidelines for the symposium before we get started. We're going to have one keynote speaker followed by 4 shorter presentations, and we'll have some breaks in between. And that's going to be followed by a 30-minute panel discussion with all of the speakers. Please type your questions into the Q&A box. And if they're directed towards a particular speaker, please indicate their name or their names. So I have a question for Dr. Abecasis, for example. Please indicate if you like a question, which helps us to prioritize which questions to address. And then we'll read some of these questions either directly after the individual talks or during the 30-minute discussion period. The speakers may also respond directly into the Q&A box for questions that are directed specifically towards them. However, we want to leave some time to address many of the broader questions as a group. A recording of the symposium will be available at the GGHE website. And again, please tweet about the symposium because we really want to make our voices loud about the need to include more diversity in human genomics research and to have a more diverse workforce in human genetics, because the two go hand in hand. And I think that's something that I'm sure is going to come up today, either in the talks and or in the discussion. So with that, I want to introduce our first keynote speaker, Dr. Gonçalo Abecasis. Dr. Abecasis serves as Vice President and Head of Analytical Genomics and Data Sciences at the Regeneron Genetics Center. In this role, he oversees statistical genetics and quantitative data sciences, driving novel genomic discovery at Regeneron. He and his team conduct discovery research in support of new target discovery, genomic translation follow-up that cuts across all therapeutic areas. Before joining Regeneron, Dr. Abecasis was, and I believe still is, affiliated with the University of Michigan, and he ran a number of their genomics programs. And he led a group of scientists developing statistical methods, computational algorithms, and software that facilitates quick and accurate analysis of genetic studies of human disease. He's also made key contributions to many important large genomics initiatives. So the TopMed initiative, you're going to hear about more of that today, the 1,000 Genomes Project, and a project that he's helped establish at University of Michigan, Genes for Good. His studies and those of other scientists using his tools are enabling a better understanding of human genetic variation and its role in disease biology. In 2014, Dr. Abecasis received the Kurt Stern Award from the American Society of Human Genetics for his contributions to the analysis and understanding of complex human traits. And in 2013, he won the Overton Prize from the International Society for Computational Biology for the tools and methods he developed for the analysis of increasing large data sets. He has over 250 scientific papers and is, in fact, one of the most cited scientists in the world. And with that, I would like to turn it over to Dr. Abecasis.
Thank you so much for inviting me. So flattered and such an important topic. Looking at your list of speakers, both for today and tomorrow, I feel a little bit inadequate to kick things off. But I guess in a little bit of the hope is that it can only get better and more interesting as you proceed through the day. I wanted to chat a little bit about just observations over time in human genetics and how they speak a little bit to these issues of diversity and equity that have really been at the forefront of all our minds for the past year for sure, and hopefully for longer. I always like to start by just saying a little bit about what brings me to human genetics, and it's different for everybody. There's many different things that genetics can do, from inform and help us understand our path and past history to helping us in some cases I understand our recent history by finding family connections. For me, the really interesting opportunity is to learn about biology, to understand how each of us works, and hopefully that with that understanding that we'll be able to develop better medicines, new interventions, new treatments. One of the things that's really important is that even though we all have the same sort of biological plan overall, with very, very tiny variations as we grow across individuals. It's what we see in our health is a combination of both that plan and the tiny variations and our environment. And I'll show hopefully some examples where it really speaks to how important that is. And obviously, Sarah just alluded to one observation that many of you are aware of, which is, just the huge difference in the impact of COVID across different groups. One of the things that has happened in human genetics, over the time where I've been fortunate to be involved, is that, the level of detail with which we can study genetic variation has just increased, really dramatically over time. In the early 2000s, a typical study would look at, hundreds of individuals, initially only at a handful of genetic markers, so you had to be, really lucky or clever in deciding which markers to study. If you picked markers in the right gene or the right region of the genome, there might be interesting things to say, but most of the time, we really weren't either smart enough or lucky enough to pick the right ones to study. But in the mid 2000s, we got to this opportunity to carry out genome-wide analysis, and that has really accelerated the pace of discovery. I mean, we all hoped actually that those genome-wide analysis would put to rest some of these ideas out there about really how big are the genetic differences between ancestry groups and individuals. I guess I remember some early analysis in the HapMap project where comparing individuals from different continents, it was really clear that the largest differences by far were in terms of the genes that determine skin color, and there was nothing else that really came close. But somehow, those ideas still haven't made it through all the way through the public consciousness. But anyway, these studies have grown. We have some big blind spots, as Sarah just alluded to, in the sense of, we really haven't studied a representative sample of all humanity. We've studied samples that were convenient and that had very big biases sometimes. And as this evolution has continued, we're now at a point where, we have studies that reach hundreds of thousands of samples, hundreds of millions, and I expect soon over 1 billion genetic variants, which will translate into something like one genetic variant every 2 or 3 bases in the genome. So, we're really looking at saturating the genome with variants, modifying and tinkering with all the little instructions in our genetic code and trying to understand their consequences. And before going deep into genetics and the main points of my talk, I wanted to take just a slight detour and just include this wonderful quote from Martin Luther King. And the quote is this: it really boils down to this: all life is interrelated. We're all caught in this inescapable network of mutuality, tied into a single garment of destiny. Whatever affects one directly affects all indirectly. And obviously, one can look at this quote and come up with many different insights. Martin Luther King was just a wonderful man of vision, but also of speaking and ideas. But I think I take it that there's just, if we do well for one group, it will help every other group. But if we do badly in one group, it will also affect us all, and it's a burden that we'll all carry. I also like to read from it that, that there's unexpected connections between things. And when we get things right and do good, that unexpected things can come out of it. And so I wanted to take a slight detour and talk about a couple of examples. And obviously these examples all fit very nicely together, when we have the benefit of hindsight and looking back in time. If you were looking forward in time and seeing them happen in real time, at least I certainly would not have predicted these outcomes. But so one of the first genome-wide association studies that I was involved in was set in this little valley in Sardinia. There's a set of 4 neighboring villages there: Lanusei, Il Bono, Eleni. And they have a total population of only about 10,000 individuals. As it happens, some colleagues had enrolled about 60% of the population in a genetic study. And so, and they had measured them for several hundred quantitative traits, mostly related to aging. One of the attractive features of the population was that, we had such complete families and often large extended pedigrees that we thought, this would be a very efficient setting for a genetic study, where at the time we were, limited in our ability to genotype large numbers of individuals, because we could pick, select individuals in these large families, genotype or sequence them, and then infer the genomes of their relatives. And so this would be a way to kind of potentiate and expand our sample size. And so we carried out these experiments around 2005 to 2007 initially. And, this is a team of several of the colleagues that were involved, Francesco, David, Serena, Carlo, Manuela, really, really fantastic people. I like to joke that, when we started, all these methods for generating data at scale were still evolving, and the papers that you see on that table were actually printouts of, little bits of DNA sequence that we had to manually review and adjust for errors at the time. But one of the early findings from this study was a look at genetic variants that were associated with fetal hemoglobin levels in adults. And so in that middle plot there in red, you see the results of one of these early genome-wide association studies. For those of you who are familiar with these plots, you'll obviously see that you have all the chromosomes laid out from 1 to 22, and then the X chromosome. You have evidence for association summarized as a p-value, and there's a whole mass of hundreds of thousands or now millions of variants being tested at the bottom, and a few peaks that stand out. In more modern versions of these plots, there's a number of extra visual aids. People will often alternate the shading between chromosomes and things like that, so it's a little bit easier to read. But the interesting thing in this initial look, and I think in many ways we were just lucky. This was one of the hundreds of traits that had been measured. Was this signal on chromosome 2 on top of a gene called BCL11A. BCL11A is a transcription factor. We didn't have a very clear idea of how you would get from BCL11A persistent of fetal hemoglobin levels. But we did have, through a couple of colleagues in the study, and I claim no part in this amazing insight, the idea of looking a little bit further, looking not just in the sample in the San Jose Valley where we started, the bottom row of that table, but saying, what would happen to individuals who had beta thalassemia, who had null beta globin alleles, and would it matter if they had what their BCL11A genotype was? And the observation was that when we looked at thalassemia major patients, these are beta globin null homozygotes who have a severe phenotype and need regular blood transfusions, we saw these BCL11A variants at about the same proportion as in the population. But when we looked at thalassemia minor patients, these are beta globin null homozygotes that actually have relatively normal lives, do not need these regular transfusions, and have a very mild phenotype despite missing the functional beta globin gene as well. And we saw, there it was much more common to have these BCL11A variants that keep fetal hemoglobin turned on. So one first hint that this gene might rescue might rescue beta globin null homozygotes from thalassemia. And Sardinia and a few places in the world, actually including Africa, are probably one of the very few places where you could have carried out this study and made these observations, because beta globin null homozygotes are not common in most parts of the world. Obviously, for those of you who followed this story, you'll know that from these sort of early initial observations, corroborated in a couple of other papers, I flagged here in the corner one by Stefan Menzel and Guillet 10 and colleagues, also pointing to BCL11A. Stuart Arkin and his colleagues really were interested in this finding and carried out some really seminal functional work to show that this was involved in, turning on and off the fetal hemoglobin gene. And now, just in the past month or two, some reports coming out, with a technology that certainly did not exist when we and others made these observations in the late 2000s, that actually, these offer hope not just for thalassemia patients, but for sickle cell patients, because through gene editing, you can, if you turn off BCL11A, you turn on fetal hemoglobin, and you can rescue many of the sickle cell phenotypes and consequences in patients. And obviously, it's very early days, and the big question is, what comes ahead. Will we be able to make these technologies really accessible and useful at scale? But with a study in Sardinia in a small set of villages carried out for reasons of convenience, coming up with this insight, having other groups follow through and do the biology, and now seeing that it's being used in therapeutics. I think it's kind of an amazing thing to see these things come together. Another example, closer to my current role, I have several colleagues that have been, working almost around the clock through the past year, trying to develop these antibodies that can help treat COVID-19 if given early on in the course of infection. And what they do is that they block interactions of the spike protein with the host cells, make it hard for the virus to spread and infect new cells. The program actually started in January, and by March, for a combination of experiments in the mouse and by looking at COVID-19 survivors from Asia, they identified several hundred candidate blocking antibodies, then went through a process of evaluation, finding the most potent ones, finding ones that bound to different parts of the spike protein so they could be used in a cocktail and make it harder for the virus to escape. Then started human testing at scale in June, early trial results in September, approved for emergency use in November, and now being used a little bit, obviously not as much as certainly many of us had hoped for, and then an early publication with peer-reviewed trial results in December. And obviously one of the questions, and as we get through the talk, I hope I'll show you a little bit of data on how human genetic data informs these kind of efforts. But one of the interesting questions for me, is what made it possible to really accomplish this at this kind of speed? And it turns out that one of the things that made it possible to come up with this treatments and strategies so quickly was work that these same colleagues had done trying to figure out a cure for Ebola. Obviously, as opposed to COVID, which is affecting, so many people close to us. Ebola often seems like a very far away disease that will not affect, luckily, most of us on this call. But it turns out that the challenge of trying to find a treatment for Ebola really got us to a place of being able to tackle COVID quickly. Part of the challenge was that the first Ebola outbreaks were relatively short-lived, and so following the traditional process of trying to develop these blocking antibodies and getting ready for a medicine that could be tested, by the time the medicine was developed, the Ebola outbreak was gone. Some of the other challenges were things like, figuring out how do you actually run a clinical trial in a war zone where there's very little infrastructure. How do you get it there? How do you help get it to patients? Can you prove that it works? Obviously, as it turns out, that worked, and that was, in some ways, an experiment that might have seemed at the time quixotic and trying to tackle a very far away problem, but actually turned out to be essential to having us ready to tackle COVID now. Okay, so back to our regular programming in human genetics. So I wanted to start just with a couple of observations from the 1000 Genomes project. So this was a project that ran from about 2008 to 2015, and really tried to survey genetic variation around the world. The design that we ended up with was to try and collect about 5 populations from sort of each of 5 different major continental areas, 5 populations from East Asia, 5 from South Asia, 5 from Europe, 5 from Africa, and 5 from the Americas, and collect about 100 individuals in each of these for sequencing. when we started, there were only about 12 human genomes sequenced. So trying to imagine sequencing over 1,000 genomes, and ultimately about 2,500 was kind of a big challenge, both in analysis methods and in technology in the lab. But it all came together with a lot of insights from many different people. Obviously, one of the things that people first ask is, how much variation do you see in each genome across different classes, whether it's SNPs, short insertions and deletions, other types of structural variation. One way to summarize that is actually just, count. And overall, if you look at how many alleles that are different from the reference genome, if you will, which is about 3 billion bases in each individual, you see that, here at the top in this sort of yellowish tones are, populations with mainly African ancestry, about 5 million of these alleles, so one in every, 1,000 bases or so, if you count per haploid genome. Then on the other end, you've got populations from Europe and South Asia and East Asia with about 4.2 million alleles. And then you've got these populations in the Americas, which have sort of a range. And the reason for that range is manifold, but in part is that, these are populations that share ancestry from different parts of the world, obviously through history. And so if you come across an individual that has more African ancestry, they'll have more variation. If they have more mainly native ancestry, they'll have a bit less. There's other patterns that, better population geneticists like Sarah and others that you'll hear about today could explain better. But, there's a very clear pattern that, we start with this huge reservoir of genetic diversity in Africa, where modern humans first appeared. And as we move through the rest of the world, we lose some of that diversity. So you see more variation in Africa, a little bit less as you go to Europe and then move through Asia. And by the time, a subset of those humans came to the Americas to found the Native American population, to see a little less variation because, all these successive bottlenecks and migrations. And so that's why you see in some of these groups like the smallest number of variants per genome. You can also ask, how unique is each population? Each one of these populations and individuals will have variants that are seen nowhere else or seen only very rarely elsewhere. And so that they have some lessons to teach us. And some of the patterns you see here is that, again, in yellow, the populations of mainly African ancestry have a lot more variation than anywhere else. You see, for example, in Europe, most European populations do not have large amounts of unique variation, but a few do. And so, for example, here, that light blue bar that stands out in that group is Finland. One observation that was also kind of striking, and once you think about it, a little bit shocking even, was what happens is you start looking at different types of functional variation. And so in this graph, you start with all variants and the relative number of variants in each population. So, for example, if you look at all variants, you'll say, there's more variation in the African samples and a bit less in samples from anywhere else in the world. And as you look at different subsets of variation, the first subset there, TFBS, are variants in transcription factor binding sites, same pattern, synonymous variants, non-synonymous or protein loss of function variants, more or less the same pattern. You start now on the three boxes on the right, you see very strikingly different patterns, and it's interesting to think about what's happening there. So these three boxes, one is for variants that are known to cause disease according to the Human Gene Mutation Database. Another one, variants known to cause disease according to ClinVar. And the last one, variants are associated with human disease based on genome-wide association studies. And, just as you'd expect from all the other categories, that that perhaps you'd see more variation in an in an African genome and less in genomes from the rest of the world, it doesn't seem to hold out quite like that. In fact, it seems like maybe there's more disease-causing variants in genomes of European ancestry. And I think what this turns out to be is the result of those biases that Sarah alluded to. Most of our genetic studies of disease or genome-wide association studies are in Europeans, and so we can annotate and recognize those variants if they're common in Europeans. If they're unique to other populations, we can't. And so we have big blind spots when trying to interpret genetic variation outside of Europe. And this has consequences if you're trying to, diagnose a disease or make a risk prediction for an individual that has non-European ancestry. You often will just not do as well because you don't have the underlying data on what are functional variants and what are their consequences. And even worse than just missing things is that often you'll get them wrong because these studies are so small and incomplete outside European ancestry that you'll just be more prone to mistakes and to making inaccurate predictions. Another side point that I like to make when mentioning the 1,000 Genomes project is that I mentioned that it was a big challenge figuring out how to analyze and interpret data at the time. And so when we started, we said, look, we'll all have the data available. Let's encourage everybody to come up with the best way to interpret this sequence data we're collecting, and then we'll do some follow-up experiments, some bake-off exercise, and figure out what was the best method among these. And so, for example, in one of the early 1,000 genome analysis, we had an analysis of all the variations submitted by colleagues at the Broad, one from colleagues at Michigan and one from the Sanger Center in the UK. And we did carry out these follow-up experiments and try and evaluate them. And the answer, if you want a very short version of it, was that, for example, the Broad had the lowest error rate, that's 0.6%, at sites that we tested and that were, in the test result should have been homozygous reference. The Michigan data had the best result at heterozygous sites, and the Sanger data was best at homozygous non-reference sites. And this actually made it tricky to choose, and we had discussions along the lines of, colleagues at the Broad would say, almost every base in every genome matches the reference. So the minimum standard is you need to get those right. You need to use our results. They're the best by far. And then, we would argue for Michigan, hey, the most interesting variants exist in heterozygous states. You should take our results for sure. And the Sanger would say, hey, the most hardest bits of the genome to call are these ones where both sites differ from the reference. You should take our results. And these are all very intricate models developed by, big teams of scientists sometimes. And, we were stuck for a long time. And actually, it took, one of our colleagues from the funding side who was very frustrated with all these discussions that never got to a decision and always said, let's collect a little more data, see if we can decide. He said, you have these three solutions. Why don't you just let them vote? If two of them agree, go with that. And if, and just vote at every position in every genome and keep going through. And I remember thinking, how could you let the two wrong results outvote the correct one? But, since they were our major funder, I said, oh, that's a very good idea. We'll investigate and see how that pans out. And the result was that, taking that very simple majority consensus, just letting the different methods vote was better in every metric. And so sometimes, instead of just having this idea that there is a single best answer or a single best mathematical model that we can build, we just have to recognize that this diversity of experiences and ideas that we each bring, if we can figure out how to bring them together, will really make for better science. And obviously this is maybe a bit of a geeky example, but I think it pans out in many other aspects of science. For the next few minutes, I wanted to just, Sarah asked me to include this and to give a shout out to the TopMed program that NHLBI started back in 2015. This was a program it's still ongoing, to advance what we know about heart, lung, and blood disorders. The idea was to take a variety of high-quality studies with really good data about outcomes related to these disorders, and now had, really good genetic data, add really good omic data about protein levels or gene expression and so on, and that the data would be regularly deposited and made available for many, many people to analyze. If you go look in that website, as I did, you'll see that as of now, there's about 168,000 genomes that have been sequenced. It's really great data, almost 40x average depth, covering most of every genome. Quality metrics look terrific. And one of the things that was unique about TopMed is that, it was a study that was designed from the ground up to be kind of inclusive, and obviously I think it's possible to do even better, but compared to the graph that Sarah was showing you, where super high proportion of samples are of European ancestry, you're seeing here that when you break down TopMed participants by ancestry and ethnicity, there's 60,000 European ancestry individuals, almost 50,000 African ancestry, 23,000 Latinos, 13,000 Asian, and 6,000 of unclassified ancestry for the purpose of this pie chart. But it's really getting closer to having that goal of being representative. And I think you could easily make the case that there's even quite a bit more to go in that direction. But I think it's really important to have done this and to show that it can be done. I think it should be, a continuing goal for all of us. Just very briefly, I wanted to highlight one of the figures from the TopMed paper that shows the distribution of variation, both by portions of the genome. It's separated here in coding and non-coding variation. And you have, in sort of darker shading at the top, the number of common variants, and in very light shading at the bottom of each bar, the number of rare variants. And these are segregated by coding and non-coding, and then by whether they seem to be, deleterious or not, based on a CAD score. But the short message is that the bulk of variation is very rare. In the table on the left, you'll see that for most classes of variants, about half of them are even seen just in a single individual. And this is why, if we want to take advantage of this opportunity to finding, hundreds of millions or billions of genetic variants, if we want to connect them to function, it probably will take, very, very large studies. But the other figure that I find striking is this one from Daniel Harris and Michael Kessler, that took all these TopMed studies, tried to summarize something about their ancestry, something about the number of variants that they carry, and then drew these little connectors. And the connectors are trying to say how many of these rare variants are shared between a pair of studies. And you see that they fall into these groups, and the groups are basically ancestry. So rare variants that are present in European studies are often present in other European studies. Those that are present in Latinos are often present in other Latino studies, and those in African ancestry individuals are often present in other African ancestry studies. But one consequence of this is that, if you could do as many European ancestry studies as you'd like, you'd have huge blind spots on variants that are not common there, and so you really need to have a good pool of diverse studies to have an opportunity to discover all that you could, and I think that in the last few minutes, I want to also emphasize the point that it's not just about maximizing scientific discovery. It's also that you can really get things wrong if you're not inclusive in who you study, because other things besides genetics can have very big impacts in our health. And so I think I don't want to run over time, so I'll be I'll try and speed up a little bit. This is just to say that the TopMed data on variation is available. You can go find this Bravo browser. It'll take you a second on Google. One of the nice things from TopMed was that with a very big set of reference haplotypes, it's possible to impute genotypes and to really improve the analysis of every human genome. And one of the things that happens when you have a diverse set of samples is that those improvements can apply across a variety of ancestries. These graphs show accuracy of imputation for individuals of African ancestry, Native American ancestry, East Asian and South Asian, and they show in blue results with current TopMed panels, and then in green and in red results with previous panels, either 1,000 genomes or the HRC. And I think the short version of this is that if you take an off-the-shelf genotyping array and apply imputation, you can recover variation in many of these genomes down to a frequency of 10 to the minus 3 or 10 to the minus 4, 1 in 1,000 or 1 in 10,000. And then this TopMed imputation server, I think at the moment about 14 million samples have been imputed using this panel. So these are 14 million samples where the analysis will be more complete and comprehensive by pairing it with these sequenced genomes. All right, I wanted to now spend a little bit of time talking about some of the things that I've observed and that keep me awake and busy in my current role at Regeneron. And so, at the Regeneron Genetics Center, we're really trying to, through a variety of studies in many different settings, sometimes working with health systems, sometimes working with family samples, sometimes working with unique populations, get a good understanding of human health and generate ideas for what new treatments or new interventions might look like. Part of the work is just figuring out how do you do these things at scale. So currently we are sequencing over 500,000 samples every year, and so my colleagues there have really come up with a fantastic platform with robotics to do the sample prep and the sequencing and proceeding with analysis on the cloud, so on. And and part of it is making sure that you can analyze this data the right way. And one of the wonderful things that has happened in the past year, through the work of Joelle and Jonathan, picture there at the bottom, was this improvement in how we could analyze very large sample sizes. For the last 10 years or so, it's been very clear that the right way to do these analysis on large-scale genetic association studies is to use these mixed models where you test one variant at a time, but you use the rest of the genome, often through a genetic relationship matrix, or more lately with some ideas that came out of Bolt and Sage using these genomic predictors to control for what's happening everywhere else in the genome, and that gives you a very well-behaved analysis. But previous methods require you to load that genotype matrix into memory in full for the early steps. And so they get really strained as you get to hundreds of thousands of samples. And, Joelle and Jonathan figured out a way to really do this, even while only loading small bits of the genome into memory at a time, which then makes it practical to, for now do analysis of hundreds of thousands of samples, but eventually for millions. And I think their work is either just published or soon to be, and you can certainly find it on bioRxiv pretty easily. This slide shows that it works well even in challenging settings where there's very small numbers of cases, very large numbers of controls, and we're looking at rare variants, just as well as Sage showed could be done, but in faster and with less memory. I'm going to skip over this for reasons of time, and spend a couple of minutes on this. UK Biobank sequencing. This is a collaboration between UK Biobank and several pharmaceutical companies, where over the course of the last few years, we've now sequenced pretty much all UK Biobank participants, and the data is going to be available to everybody who's an approved user of the UK Biobank data set. Obviously, it's kind of amazing to look at a data set of this size and with very complete health data to see that we can recapitulate many of what's known about human genetic diseases, across a range of settings. You could find, for example, associations between PKD2 variants and kidney disease, and these are homozygous and heterozygous counts in this for this particular gene. There's only 15 plus 24, about 39 carriers in the whole of UK Biobank, or at least in this portion of the sequencing, and out of those 39, 15 had polycystic kidney disease. So that's an odds ratio of about 1,000. And then as you go through, you'll find that there's a range of genes with a range of effect sizes. These are just selected examples to make the point. At the other extreme, you have, for example, variation in filaggrin, increased risk of atopic dermatitis, but only by a factor of about 2, and with many, many things in between. You can also take, any gene of interest or any variant of interest and try and understand what's its impact across many different traits. For many people, a favorite variant is this variation in ApoE. It's associated with cholesterol, as you see here at the top. It's also associated with Alzheimer's and dementia. In the case of UK Biobank, there's a stronger association with your parents having dementia than with the individual themselves having dementia, because it's still relatively, there's still a relatively young cohort. Not many people have developed Alzheimer's or dementia yet. Many of you will also recognize ApoE as one of the genes that we always think is the prototypical example of a gene that's associated with aging. And so I wanted to just take a moment to show you a very, to me, very, very striking result that really speaks to, even if we think biology and genetics are shared, if we don't study, the right people, will have completely different answers, and we could miss things, we could be misled, or we'll certainly be incomplete. And so this is, for example, looking at what impacts age of death in women in UK Biobank. And in this case, as a proxy for that, we're looking for each participant at what age did their mother die. And it turns out that you see ApoE, as you'd expect, and then you see two other hits. One is the cumulative effect of genetic truncation variants in BRCA1 and BRCA2, and each one of those will be associated with a lifespan of about 5 years shorter for your mom. ApoE, in contrast, is much more common, but only about a 3-month shorter lifespan. That's, to me, pretty striking. And what's even striking in comparison is what happens if you look, for example, at father's age of death instead. So again, looking at longevity, but just, very similar people otherwise, except in one case, a group of all women deaths, and the other case, a group of all male deaths. And ApoE is there, but everything else is different. So here we're seeing genes associated with propensities to smoke, genes associated with cardiovascular disease, genes that appear to be determinants of immunity. And so it's really kind of striking that for a trait that, superficially we might have expected to be a bit more similar, that we have ApoE there in the corner is shared between them and with similar effect size. And then we have all these other towering peaks that are unique to each sex. And when you think about, any condition where there's big environmental effects or big effects of socioeconomic status or diet or where you are, all things that we know are correlated, with ancestry and diversity. You could easily have very different results if you study just one group of people as opposed to being inclusive. Obviously, there's other kinds of blind spots that are just about, do you have enough variation? And this one is the example, for example, PAX4. We have a study that's ongoing in Mexico City, where we've now sequenced about 100,000 individuals from the Mexico City population study. And in that sample, you can really straightforwardly recover association of, for example, PAX4 with diabetes. And this is an association that others have described as important in individuals of Mexican ancestry. But in UK Biobank with 4.5 times as many individuals, you really would not find this signal because the variant is too rare there. But I think we should not obviously it's important to see the variant, but we should also not discount this issue that the things that matter to health in the end are not all genetics, and so we really need to study diverse groups to capture all of them, and ultimately to understand biology in all these groups and be able to come up with treatments and interventions that benefit everybody. Otherwise, people are going to be left behind. I think I'm a little bit stop here.
You're okay, Gonçalo. You can go on for another minute or so. That's fine.
Okay, I was just going to mention that another study that's been really interesting for us over the past year has been to try and figure out, could genetics contribute to identifying and understanding, COVID. We have a paper in bioRxiv that includes a meta-analysis across about 20,000 cases. 7,000 of these are hospitalized. The same data has been contributed to this terrific human genetics initiative effort that's ongoing, that's trying to aggregate an even larger sample set. And I wanted to just make two really side points. Obviously, genetics does point to very interesting modulators of severity and infection. But in each one of these data sets that contribute to the analysis, whether it's the one we generate with terrific colleagues at Ancestry or UK Biobank or Geisinger, or actually, Sarah, with some of your colleagues at Penn Medicine, we see this, big excess of, cases that have non-European ancestry. So, for example, in Ancestry DNA, 86% of the people in whom we have data have European ancestry, but by the time you get to hospitalized cases, they're only 3/4. And as you go to more and more severe outcomes, it's a pattern that's repeated again and again, and often the discrepancies are even bigger than that, than in this particular example. I think another thing that's been really striking is that, for example, if you stratify clinical risk factors, things like age and comorbidities and so on, in those first two blue bars, you can identify very high risk groups of individuals, a bigger chance of severe disease if they're infected. You could take the genetic variants that define the peaks of those genome-wide association study plots, and come up with another set of risk factors, and you'll see that again in gray and red there's a clear pattern. But the really interesting thing is that they seem to be almost independent. When you combine the two, you can stratify the high clinical risk group and find individuals that are at even higher risk. You can stratify the low clinical risk group and find individuals that have high genetic predisposition and they don't have many other risk factors, but they have almost the same chance of being hospitalized and getting severe disease as those that had just the clinical risk factors but no genetic risk. I wanted to say, obviously there's a lot of work from many different people that goes into, these summaries and stories that I'm telling. And so I just wanted to give a shout out to all of them and thank them. And then I think I'll end here if that's all right.
Yes, thank you, Gonçalo. That was an awesome presentation. We can take some questions that were in the Q&A. And I see that some of them are related to each other. So I'm going to ask you two questions that were submitted. So one is the question, is the proportion of each ethnicity in the TopMed model proportional to the number of individuals representing these ethnicities in the USA? But then the second question said, or comment, is sampling proportional to demographics enough to produce equitable outcomes in research? So for example, all groups receive the same benefits or level of information from the research, or would we need to oversample minority populations or groups?
So I think those two questions have answered each other, so I think it's really important to oversample. If you think that, every life is worth saving, we can't say, look, for you guys, there's not that many people like you, so we didn't get around to studying, so we don't know how to help you. So it's important to get large numbers in each of these groups so that you can actually, learn what you need to be able to come up with intervention. Sometimes just understanding, so I think, it makes a lot of sense to oversample, underrepresented groups.
I agree. One group that I saw was missing are American Indians. And I'm wondering if that might be something to do with the challenges of doing genetics research in this group. And I know that's going to be a focus of tomorrow's symposium, but I don't know if you have any comments about that.
Yeah, I mean, I think when we have groups where we, as researchers, have not always done the right thing, we make it very hard to then say, now we're new people, we're going to do the right thing this time, come participate and share your data like everybody else. I think those messages are hard, and it just takes a lot of work and doing the right thing over and over till we get there. I think it's a challenge of enrollment. It's a challenge that we haven't generated the sample resources as much as we have for some of the other ancestry groups. It's clearly one of the places where I know the folks who are leading TopMed and trying to organize it are keen to do better, but it's hard for many different reasons.
So that totally tied into the third question someone asked about, do we need to be allocating more resources? And clearly you've said for studying minority populations, and you've said, yes, we do. And that's something perhaps we'll hear more about from Dr. Gibbons and others. So one question is, what are the advantages and disadvantages of correcting for ancestry in rare variant association studies versus common variant association studies?
Okay. So, look, I think in each of these studies, you want to make sure that you're learning about biology. Sometimes genetic variation tracks with ancestry, and so it tracks with all the other things that come along because of ancestry. So if you want to be able to say, I discovered that this gene changes your risk of glaucoma or changes your risk of cancer or getting hospitalized with COVID, you want to know, you'll learn different things if that gene is acting because it worked through skin color and it subjected you to discrimination or something like that, versus it's a biological signaling mechanism that made it easier for the virus to come in, or for your made your vision degrade faster. And so we really just need to separate, both in common variant and rare variant studies, what are things that are due to ancestry overall, and what are things that are the consequences of specific genetic variants. And it sometimes is a hard problem. It's why it's important to think about analysis methods and put them to the test, and try and interpret these results, not just put out a genome-wide plot and leave it there.
So I was curious, I noticed you mentioned the PAX4 association that you found from participants in Mexico. Is that right? But you did not see it in the UK European database, is that correct?
That's right.
And could there be is that because these variants are only in those populations and they're not common, I guess, in the European population? Because of course at GWAS depends, right? You're not going to see a significant association unless there's some kind of variation. But I'm also wondering, could there be a gene by environment effect? So could there be a case where the variants are there, but you're only seeing the phenotype, let's say in a Mexican environment, something like that?
Sure. It certainly could be both. I think right now the parsimonious explanation is that we can't tell what's happening with those variants in the UK because we don't have enough examples. It could be that with more samples, the association would show true. It could be that, with more samples, hey, actually, this doesn't show up there. It's something about everything else that's happening that's different between the UK and Mexico.
Okay, I'm just going to ask one. We'll just do short answer if you can, then we got to go on a break. If we have moderately large sample size per ancestral group, is it preferable to conduct analysis stratified by ancestral group as compared to a trans-ancestry analysis for a given phenotype or disease?
This is where you ask one statistician and you get two answers, and the answer is it depends. I think we have super lively discussions about what's the right way to do this internally. I think you can find examples both ways. When and there's examples where you probably want to do neither of the above. You want to look at each group separately because the genetics are just so different. Like imagine looking at that age, males versus females, and just meta-analyzing it. If anyone's sort of super pressed, I really want to help figure these things out, I know they could reach out to you. I know they could reach out to us. We're also always hiring and looking for smart people. So, but that's my last bit.
I'm going to go ahead and stop there and say we're going to go. Thank you so much again, Gonçalo. I'm sure if everybody was here, there'd be lots of loud clapping. Ricardo, I want you to know I saw your question, but I'm going to save it. I think it's actually good for the general discussion. I'd like to welcome everybody back and introduce our next speaker. And just to remind you, in this next set of speakers, they're going to be 20 minutes each with a few minutes for questions. All of these speakers have amazing backgrounds. I wish I could give them long bio introductions, but because of the short time, I'm mainly just going to be giving names and titles of talks. So our first speaker is Marylyn Ritchie, who's professor of genetics at UPenn and director for the Center for Translational Bioinformatics and associate director of bioinformatics at the Institute for Biomedical Informatics at UPenn.
Thank you so much. And thank you to Sarah and Dan for inviting me to participate in this tremendous symposium. I'm really looking forward to it. So today I'm going to talk about polygenic risk scores and whether they are for some or for all. I'm going to start with my conclusions, which you'll see again at the end, but I want to make sure that there are 3 points that you're keeping in mind as I talk through the material today. Polygenic risk scores, which I'll define them for you on the next slide, or over the next few slides in case you're not familiar with them. But they're a technology used in human genetics, and they look promising. But we have a lot more work to do in order to take this technology and make it not just for some, but for all. For them to be useful in precision medicine, there are 3 essential issues, and there are probably 30 other issues that I could talk about, but I only have 20 minutes, so I'm going to focus on 3. Number 1, and the one that I'm going to spend the most time talking about today, is ancestry. And it's both global or continental ancestry as well as local ancestry around particular genetic variants. And I'll show you more about that later if you're not familiar with local ancestry. This ancestry information is critical to have accurate estimation of risk. Ignoring this, I think, is going to cause huge detriment to this technology of polygenic risk scores. Number 2, and this is something that Gonçalo somewhat mentioned throughout his talk as well, it's all the other factors that go with genetics. So clinical, behavioral, environmental risk factors, and social determinants of health. For most polygenic risk scores right now, they're not incorporated into the model. And I think it's essential that we figure out how to incorporate those with the genetic background to estimate risk. And then finally, what I'll end with is that equitable implementation of polygenic risk scores into clinical care must be required. The equitable part, we have to do this in an equitable way to reduce health disparities. And anything short of that is only going to exacerbate and increase health disparities. So first, let me describe polygenic risk scores for those who are not familiar. I'm going to call them PRS, or polygenic risk score today, but if you look in the literature, they're also referred to as genetic risk scores, polygenic scores, genome-wide polygenic scores. There's a lot of acronyms that mean essentially the same thing. It's essentially an aggregation of genetic variants into a score. So you start with a genome-wide association study. And Gonçalo mentioned this in his talk as well, but this is the basic concept where you have cases and controls, a group of people with and without a particular disease. You run a genome-wide association study where you look for the association of each variant across the genome for the trait, and you end up with your kind of summary statistics. Here, this is a Manhattan plot of chromosome position across the X, negative log of the p-value on the Y. The summary statistics, or the betas that go with this GWAS, are what move forward into the polygenic risk score. So you take those summary statistics along with LD reference data, this is linkage disequilibrium or correlation across the genome, along with some PRS method. And what I'm showing here is a very simplistic PRS method. I'm not going to get into the statistics of how these work. One, because statistics are often boring in a forum like this, but also there are about 15 to 20 methods that we could talk about. No one method has been shown to be superior to all others. So this is an active area of research. But the basic idea is that you take the summary statistics and LD reference data to estimate risk in an independent study data set, and you'll create these distributions and identify who's at low risk, who's at intermediate risk, and who is at high risk for a given phenotype. Now, I would postulate that we really need to be thinking about ancestry all steps of the way. The ancestry of the GWAS data set for which those summary statistics are derived, so those betas or those effect estimates, we need to know the ancestry of those individuals. They should be similar to the ancestry of the LD reference panels that we're using. These are those correlation structures or haplotypes across the genome. If we want to look to see how that genetic signal exists along a chromosome together, we need to have an LD reference panel that is similar to that summary statistic data set. And both of those should match our independent study data as closely as possible in order to get accurate estimation of a polygenic risk score. I would also add that in this PRS methodology, there's a variety of ways that statistical geneticists are integrating this LD information along with the summary statistics to generate these risk estimates. And the that we can do to use this LD reference data, as long as it matches our study population, the more accurate we are likely to be. Now, why is this so important? There have been a lot of publications in this space, and I'm going to highlight just one of them because I think just this one image helps to really get the point across as to how it can be a problem if you don't have the right type of ancestry all along the way. This study, which is led by Emer Kenny at Mount Sinai, looked at 5 different global populations for a variety of traits. I'm only going to talk about 2 here. But what you can see is that in the polygenic risk scores for height, the risk score would estimate that, so Europeans are represented here in this blue color. And for example, African ancestry, which is here in red, is estimated to be lower, which indicates that based on the score, Africans would be on average shorter than individuals of European ancestry. That is simply not the case. If you look at global data across European and African ancestries, heights are very similar. And so here we have an underestimate of what the actual height would be. Similarly with schizophrenia, where the prevalence of schizophrenia is actually very similar in individuals of European and African ancestry, again, we see that the risk is higher in Europeans and it's estimated as lower in African ancestry. And that is simply not the case. It's because the summary statistic data that was used here was kind of preferentially of European ancestry. And so the risk estimates that it's producing are underestimates for the African ancestry individuals. There are also examples in this paper, and if you're interested, I encourage you to go look at this. This is now 4 years old, but those estimates show that you can also overestimate risk. So you could say that individuals in other populations are at higher risk because you used summary statistics that were not appropriate. And so we don't want to make these risk estimates inaccurate for different populations. The whole point is to predict risk in an efficient and an accurate way so that we can then do preventive measures or different treatments to, maybe prolong the severity of a disorder. And so we want these estimates to be accurate. We do see in both simulation and real data applications, and there are a number of these in the literature, that you get optimal performance if you include diverse cohorts in the summary statistic, the LD reference panel, and the study data. So you might be sitting there thinking, well, this is obvious, and why aren't we already doing this? Because this just seems like a natural thing that we should do. And I'm going to highlight a paper that Sarah Tishkoff mentioned earlier, and she's the senior author on this paper. She talked about this figure on the right, the number of people represented in the GWAS catalog. But I'm going to focus on the left, the studies in the GWAS catalog. These are the studies that we're using to get those summary statistics to take into PRS. They are half European. And if you look at any other continental ancestry group, they're much, much smaller. And so part of the reason that we are not doing this well currently is that the summary statistic data sets either don't exist for the phenotype of interest, or they're just so small that the effect estimates are biased and not very accurate in the GWAS. And so then taking those forward only propagates that inaccurate estimate kind of down through the PRS pipeline. And so we really need to increase the number of studies in diverse groups to build the beginning of that pipeline, the part where we're estimating the summary statistics. There's a great paper by Barbara Bittarello and Ian Matheson here at Penn that really dug in on height specifically in admixed populations. And this is where I'm going to transition into this local ancestry idea as well. I want to highlight 2 sentences from their abstract, and I really encourage you to look at this paper. So one statement, they showed through a number of analyses in this paper, it's really well done, that recombination rate, which is the rate that the chromosomes recombine, differences in allele frequency, and differences in marginal effect size across ancestry all contribute to the decrease in predictive power of the PRS across ancestry groups. But none of those factors were explaining the decrease on their own. So it's a combination of factors. It's not as though if we just had the allele frequencies right, it wouldn't be a problem. It's all of these things in combination that lead to the scores being inaccurate. And secondly, they demonstrate that prediction in an admixed population, so this is where the individuals have a combination of ancestral haplotypes from different regions of the world, can be improved by using a linear combination of PRS that includes the ancestry-specific effect sizes. This is currently not done very often. Again, back to the earlier point that we simply don't have the data. The sample sizes are too small and then lack power to do this effectively. I saw this great paper that came out last year that really dug into this local ancestry idea. And so, as I mentioned, I'll explain it a little further. In an admixed genome, it is not the case, and I should take one step back. Even in a European genome, an African genome, any kind of continental ancestry classification, we are not homogeneous. It is not as though for most people currently alive in the world that every part of our chromosomes were derived from the country of origin. There's certainly a lot of mixing across different countries within different continents. But even more so when you think about individuals where the ancestral chromosomes come from different countries of different continents, what you can expect is that along each chromosome, it's almost like a painting of different haplotype blocks from different regions of the world. And so if we don't consider that when we're building these PRS scores, where, for example, if you take these chromosomes that are a combination of red and blue and gray and break them down into these two different groups. So, for example, in this region, an individual comes from one continental group, and in this region, it's a different continental group. Well, if the genes that are important for inferring disease risk are in one of these kind of haplotypes, but not in the other, and we just classify this individual either by self-identified ancestry or by their kind of highest proportion of ancestry, that's actually not relevant if the regions of interest are from a different ancestry group. And so when I saw this paper, a light bulb went off. Like, this is the type of methodology that we need that really teases apart local ancestry and then brings those together in the polygenic score, such that we can look at the effect estimates in the GWAS summary statistics in the regions for which we have different ancestral haplotypes and then build the score that way. The method that they used I thought was interesting, but I would say, you can insert a lot of different methodologies here, and I expect that in the coming years, we're going to see a lot more efforts put right here. In order to do this well, we need more diverse data sets to derive the summary statistics. We need more diverse data sets to derive the LD patterns, and we need to improve our methods such that we take this local ancestry into account. All right. The second area that I think it's really important that we work on is incorporating these other factors with the PRS. Clinical, behavioral, environmental, social determinants of health. How can we do that in an effective way? And this includes family history as well as monogenic risk, such that we have more accurate risk predictions. As Gonçalo mentioned so well in the last talk, we know that it's not the genetics in isolation, it's the genetics in combination with our lifestyle, our environmental exposures, our social factors. So how can we put these together? And this is something I would encourage all of us to keep an eye on what the NHGRI eMERGE4 network is doing. This is a figure from their website. This is exactly what that network is poised to do over the next several years. And I think a lot of us in the field are working in this space of what I would call integrated risk assessment models. So in a lot of disease areas, we have clinical risk scores. How do we marry that with these polygenic risk scores and develop integrated risk scores that we don't just want to adjust for clinical risk or adjust for social determinants? We want them to be part of the modeling to have accurate risk prediction. There are a lot of biobank resources, both population-based and EHR-based, electronic health record-based, all around the world. I just found a small set of logos, just of a few of them, to make the point that I would predict that it's these types of resources that are really going to enable this to happen more quickly, because these types of resources are collecting certainly clinical information. So all of these that have electronic health records have a lot of clinical information available. However, we are learning that the clinical data can be biased just because of healthcare utilization due to systemic racism and bias. And so we need to do a better job of collecting more information from the research participants and not relying simply on the electronic health record alone. So from the EHR, we can get things like diagnoses and clinical lab measures and imaging. We can collect biospecimens and do genomic analyses on them. But we need to think about creative ways of engaging participants to get the rest of the data. Things like surveys where they could answer questions about sleep and physical activity and nutrition. Wearables are becoming very popular, and I've seen a lot of studies where they're giving wearables away to research participants to track, how much physical activity are they getting or how much sleep, rather than people having to remember or report it. A device can report that information. And the other is one that we don't even necessarily know about all of our exposures, and that's through using geocoding. So if we know where people live, you can map your location to public data about your social determinants of health and exposures. So things like, if I asked you, how much air pollution are you exposed to? Do you actually know that? I certainly don't know that, but the EPA does. The Environmental Protection Agency in the US has monitors all around the country. And so you could look at where you live and how close that is perhaps to a highway or to some plant that's putting toxins into the air. And that can help us learn what some of those environmental exposures are that might influence our genetics in these polygenic scores. I think it's going to be a combination of all of this that's going to make these integrated risk models successful. And I think that leveraging learning healthcare systems, these academic medical centers and health systems that have this kind of learning health system model will really enable us to test this. So it's one thing to collect the data and have these nice diverse data sets and great statistical models, but we also have to evaluate, how do we implement them into clinical care? How do we use them so that patients are protected, so that doctors know what to do with them? And how do we think about these different patient populations like pediatrics? What do we do with people that are low risk? Do we tell them that they're low risk? I don't know that that's necessarily a great public health thing to do. And what's the psychology of telling someone that they're high risk? These are all things that we need to learn, and doing this in the context of a learning health system is likely to be really fruitful. Lastly, I just want to point on the equitable point. If we don't do this in an equitable way, we are going to increase health disparities. Full stop. This has been written about in a lot of perspectives. This is a great one by Alicia Martin et al. I strongly recommend taking a look at it, but you can find lots of these. And here is why this can become an issue. I should say, this is a screenshot from Myriad's webpage. I have nothing to do with Myriad or any other pharma for that matter, or any other genetic testing company. So this is not an attack of Myriad. It's merely an example from the internet of how I worry about using these types of scores to drive and increase health disparities. So there's a risk score for breast cancer that one can order. And in fact, you can, as a patient, click here and order now to order this breast cancer risk score. It pulls together both Mendelian, kind of the BRCA1, BRCA2, as well as genome-wide association study data to predict risk of breast cancer. This is fascinating. I didn't think that the PRS for breast cancer was that accurate yet, but clearly I was wrong, and there is a test available. And so I'm looking through the web page, and I was like, this is fascinating. I did not know this was happening. And when you get to the bottom of the page, it has the inclusion criteria, or who is eligible. European and Ashkenazi Jewish ancestry women. Those are the only ancestry groups that are eligible to order this test. Now, as a scientist, I know summary statistics are generated in European and Ashkenazi Jewish women. Therefore, there's confidence that the risk estimates are accurate in these populations of women. But the website doesn't say that. The website says this is a test for white women. This is surprising, shocking, appalling, all of the above. I actually identify as a European and Ashkenazi Jewish ancestry woman. I have ancestry that is Ashkenazi Jewish, Italian, and Polish. And so I'm eligible. I don't even want to take the test because my friends can't take the test. This is something we have to avoid. As scientists, we have to avoid doing this because currently there are polygenic risk scores for a lot of traits that look pretty good in individuals of European ancestry. But if we offer them and don't offer them to other ancestry groups around the world, we are going to take what is already health disparity issues and make them larger. So to summarize, I am really excited about polygenic risk scores. I think they have a lot of potential for precision medicine. I think there's a tremendous amount of work that we can do with them. but we have to address this ancestry issue. We need larger studies of more globally diverse populations. I think Gonçalo already said that, and I'm going to reiterate that point. We need improved methods to estimate the PRS. There are a lot of people around the world working on this, and so I think this is something that we'll see for years to come. Thinking about how to collect that unbiased data in global populations is going to be really important. The electronic health record is great. I use it a lot, but we know that it doesn't have everything. And so how do we get those other data? And then how do we integrate them into these integrated risk assessment models to really not just control for these other risk factors, but incorporate them and find those gene-environment interactions so that our risk predictions are based on the holistic picture of the human rather than only their DNA. And then there's a lot of ethics and a lot of work to be done around this equitable implementation. I think working in health systems where we can start to kind of do these research studies to see how do we implement the PRS in an equitable and psychologically safe way for the patients and providers is going to be a really important challenge for us in the coming years. So with that, I just want to acknowledge all of the collaborators at Penn that talk about these things. This is something that we spend a lot of time thinking about, talking about, working on methods for, especially the members of the Penn Medicine Biobank team, where we're doing a lot of this polygenic risk score modeling currently, and really thinking about, there was a question earlier, you know, our ancestry distribution is about 30% African ancestry and 70% European ancestry. How do we do this well so that we develop scores that work for 100% of our participants, not just the larger subgroup? And so with that, I will stop, and hopefully there's still time for questions. Thank you.
Thanks so much, Marylyn. Awesome talk. I totally share your concerns about the construction and the application of polygenic risk scores. And given the fact that they perform so poorly across ethnic groups, I'm going to ask you a provocative question. Should we even be using them right now?
No. Yeah. I don't think we should be using them right now.
I think I would.
I think we need to do more research to understand, because even in the European group, we're not taking into account the other risk factors. And so health disparities aside, I'm not sure they're ready for prime time. But if we put on our social justice hat, which we all should be wearing at this point, no, we should not be using them yet.
Okay, so somebody asked in one of your slides, in slide 6, the schizophrenia score, panel B, seems to have a much wider distribution in the Africans and was also left shifted compared to non-Africans. It seems the polygenic risk score does a better job stratifying African people. Can you comment on this?
That's not what it's doing. So what it's doing, so the reason that it's left shifted is that it's predicting that the risk is lower, which is not accurate. It's, I think, doing that because many of the variants that are driving the signal in Europeans have just a very different frequency in the African ancestry group. And so they perhaps look lower risk. The width of that distribution, I believe, is because the sample size is so much smaller and the variability in allele frequency across the genome is so much higher. As Gonçalo showed, you know, if you take any African ancestry group, they have a lot more variability than any European ancestry group. And so they're going to have more variability of frequencies, which means when you multiply by those betas, those summary statistics, you're just going to get a much different distribution of scores. And they're just much less accurate because of that.
And somebody was asking, what did you mean by ancestral weights? Was that like for the local ancestry, including that information? Is that what you meant, or global ancestry?
Yeah, so what a lot of the methods are doing right now, they're weighting, so in those polygenic scores, you have the beta from the GWAS, and then there's some sort of correction for ancestry. And right now, most of the methods use global. So are you from Africa, Europe, East Asia, Southeast Asia, East Asia, et cetera? What these local ancestry methods are doing, I think, is going kind of, sliding along the genome and putting a weight that's really based on just that haplotype region rather than your whole kind of, continental predicted ancestry.
Right. So we could go on a long time. A bunch of questions have come in, and hopefully we'll get to some of these during the discussion panel. Thank you so much, Marylyn. And now I would like to introduce our next speaker, Dr. Neil Hanchard, who is very recently now at the Medical Genomics and Metabolic Genetics investigator at the Medical Genomics and Metabolic Genetics Center at the National Human Genome Research Institute. And welcome, Neil.
Okay, so first, thank you very much for inviting me to speak, but mostly thank you for putting this together. I think this is really timely for our field, and in some senses, perhaps overdue. So what I wanted to try and do today was kind of restate the case about diversity in genomics and focus more on sort of African genomics. And I'm going to try and use the H3 Africa project to provide some context behind that. And then I want to finish off by giving two short clinical vignettes about how the work that is occurring in Africa is starting to have an effect upon individuals of African ancestry outside of Africa. So this, of course, is a slide that you'll see quite often, I'm sure, for the next couple of days. But just to restate that, using genome-wide association studies as a proxy, the vast majority of these have occurred in individuals of European ancestry. And then if you break that down even further by individuals, you can see that African individuals are a very small portion of this. And that in some ways is a very cruel irony because, as was pointed out by our keynote speaker, African populations have the majority of novel or new variation generally. And this, of course, reflects the sort of origins of modern humans in Africa and their subsequent migration to populate the rest of the world. But the important point is that in leaving Africa, not everybody left. And so there's a very deep ancestral tree, as well as a very rich tapestry of genetic variation in the continent. And so this disparity where there's a lot of genetic variation, but very little actual genomics done, was very much the genesis of what is the Human Heredity and Health in Africa Consortium, or the H3 Africa Consortium. And this is really an NIH and Wellcome Trust, and more recently, the African Academy of Sciences funded project to empower research and researchers on the continent. And the idea is to provide both funding and therefore access to sort of modern cutting-edge genomic technologies in pursuit of diseases that are burdens on the continent. But more importantly, perhaps, to develop both the physical and the human resources to enable genomics and genomic studies. And so the setup for this is this kind of hub and spoke framework where there are a number of collaborative centers, each working on a specific disease with multiple projects. There are a number of research projects that are individual. There are ethical, legal, and social issue projects. And then this is all buttressed by biorepositories that have aliquots of all samples that were recruited. And then there's a robust bioinformatics network across the continent, and that feeds into training for bioinformatics. And so now in its 9th year, you can see that the consortium has sites all across Africa. And what's important about this and makes it unique is that in these cases, all the grants were awarded to PIs who are in Africa. And that was a primary stipulation of what the project had to have. And so now has about 34 different countries. There are 51 or so projects that are supported. And through the sort of regular meetings and working groups that have formed, there are a number of policies and guidelines that have been developed to guide sort of genomic and genetic research on the continent going forward. As you can imagine, the number of individuals has swelled quite significantly. There are over 500 consortium members now. This is almost the last time that we got the chance to meet together in Tunisia. And I promise you, I am somewhere up here in the back. So currently, the number is over 70,000 participants who have been recruited. and sometimes recruited in fairly inhospitable kind of situations, either as a result of environmental factors or sometimes as a result of man-made congestion. And these projects span the full spectrum of human genetics and human disease. So there are infectious diseases, and then there are sort of non-communicable diseases, and then everything in between. And at the start of the project, there was this idea, this was the sort of landscape of whole genome sequencings that were available. And so the majority of these in the light gray are from the 1,000 Genomes Project, and then the two dark gray ones were from the African Genome Variation Project. And of course, this is centered on Nigeria, where the Yoruba and to a lesser extent the Igbo have been sort of the proxies in the broader field for West Africa, and in some cases even Africa. And so there was a general feeling that this landscape was going to be insufficient to represent the studies that were emerging, and particularly the more than 2,000 ethno-linguistic groups that have been described across the continent. And so all the principal investigators were invited to submit control samples from their projects that were appropriately consented to have whole genome sequencing done. And this resulted in a broader selection of sequenced individuals. So we were able to sequence at fairly high depth. You see in the red dots here, about 348 samples, and then a number of individuals sampled at lower depth in the green dots. And again, the important point here is we're trying to get to those populations that are not the ones that have been previously evaluated. And so having gotten the sequence, the first thing that we did was start to look at this in a broader context than here in the space of the first two principal components. And what we could see on the first principal component is that we start to separate the sort of traditional Nilo-Saharan groups, so classic Nilo-Saharan groups of the eastern horn of Africa, from the Niger-Congo groups. And so even though we're going into new groups, we're still within this big language group that includes the Bantu language of Niger-Congo. And you can see that gets separated on the first principal component. And then on the second principal component, we see this kind of geographical cline where you're moving from Western Africa through Central Africa down into Southern Africa. And again, this is not sort of discrete clusters that are separate, and there's more sort of merging of groups as you move from one region into the next. But even within this Niger-Congo cluster, we could see exceptions that told us that there's more variation there than we may have imagined. The quintessential example turned out to be the Birom from Nigeria. And so on these admixture plots, and again, the way to read these things is each line here is a different individual going each row, and each color represents a different sort of ancestral component. And you can see that in the Birom, compared to the neighboring groups in Nigeria, there is this dark blue ancestry component. And through a series of other studies, we're able to show that this is most likely coming from East Africa and most likely Chad several thousand years ago, and really indicating this is not the first description of that kind of introgression, but certainly the Birom are a fairly large group. There's 2 million individuals. And the bigger point is that, we start to think about people when people are proxying West Africa or Africa through the Yoruba. That may not even be a great proxy for Nigeria, not to mention the rest of Africa. So the other part, as we started to go into these new populations, we were able to look at how many new variants were we able to discover. And this is on top of what's available in the public databases and 1,000 genomes, etc. And what we could see is that, here we're starting with Botswana in the south, and then saying how many additional variants do we identify when we go to each new group. And you can see that, for all the single nucleotide variants that we have, there's, no real plateau approaching. But even if we said, well, let's just focus on the common SNVs, then you can see that although it looks like we're getting to a plateau here, it's a bit of the scale problem because there are almost 6,000 variants over these last two individuals from Benin and Mali. And in fact, we found that, once you go outside of this Niger-Congo group, there's actually a fairly strong correlation between the number of novel variants and your proportion of non-Niger Congo ancestry here in the groups from Mali. So again, as you go out more in Africa, you're starting to get more and more variation. So then we were interested in how we could make this sort of clinically relevant. And so one of the things we did was we looked for signatures of selection across the genome. And so we found, several signatures, 60 of which were unique and had not been described before. And they varied in their intensity from country to country and from region to region, as far as the ones that were overlapping the coding regions. What was interesting, however, is that we did find a number of loci that appeared to be under selection in the non-coding regions. And in fact, 36 of those actually had established genome-wide association hits. And those hits mapped to 24 different disease traits using the GWAS catalog. And in fact, 5 of these were actually represented more than once and represented multiple times across the populations that we looked at. What was really interesting is that when you looked at the diseases that were being represented by these overlapping hits, you can see things that are known to be quite common in individuals of African ancestry. So chronic kidney diseases, uterine fibroids, and variable red cell indices are all much more common in persons of African ancestry, and possibly also eye morphology if you think about things like glaucoma rates. Okay, so that's sort of very common and complex diseases. Then we wanted to think about, well, what about rare diseases? And you'll hear more about this tomorrow, I'm sure. But just to give us a preview, we looked first for the variants in the American College of Medical Genetics and Genomics list of medically actionable genes. And you can see that very few of our individuals carried any of those kind of variants. But as was alluded to by our keynote speaker as well, we also looked at individuals in ClinVar. We looked for ClinVar variants that were known to be pathogenic, and we found that almost everybody carried at least one of those, and most people carried 7. And this is sort of counterintuitive. You'd expect that these are very rare variants, and they're rarer in public databases, but again, the databases don't include the breadth of individuals that we had here, and so this is likely a sort of misclassification problem where you're sort of assigning pathogenicity partly due to the fact that these are relatively novel, but not so common in the African context. So finally, we wanted to look at those variants that are known to be relevant in the African context, but maybe aren't represented as well in the sort of medically relevant reports on this side. And so we did this looking across the continent, and so you see here, for instance, the beta-globin sickle allele, and you can see it's fairly high frequency as you move across sort of the equatorial part of Africa, in keeping with sort of those regions where malaria is endemic, the primary driver of the allele frequency. And we could see something similar with variants in APOL1. So again, APOL1 variants are protective from certain types of trypanosomiasis or sleeping sickness, although paradoxically, they can give you an increased risk for renal disease, particularly in the context of HIV. And again, you see this sort of variation across those areas where you have very high trypanosomiasis endemicity. But once you go outside of those regions, go further south, for instance, start to see that those frequencies are very, very different. And this is sort of well exemplified for the 2 groups in Uganda. So again, 2 groups, both from Uganda, recruited as part of studies. And there is the Bantu-speaking group, or UBS, and then the Nilo-Saharan group. And what you can see is that the frequencies of the variants are very different in these 2 groups. And this reflects their ancestry. So the Nilo-Saharan group are predominantly from, are thought to have migrated further south from the eastern horn of Africa, whereas the Bantu-speaking groups are more in keeping with our Niger-Congo groups here. And so they're very different allele frequencies, even though you're looking within the same country at medically relevant variants. Okay, so that's kind of the big picture of how things planned out. So what I wanted to do is to finish up giving sort of 2 short clinical vignettes about how that may be relevant when you start to look at populations of African ancestry outside of Africa. And so the first example is talking about genetics of iron overload in persons of African ancestry. And so iron, of course, is very important in the body. Too little iron is the number one cause of nutritional anemia, iron deficiency. But also iron overload is also a significant problem that can occur. And so that homeostatic balance of iron is very important to get just right. And red cells, because of their rich hemoglobin, are a major contributor to iron in the body, the breakdown of hemoglobin. And so Ma and colleagues in a recent issue of Cell showed in a mouse model that this gene PIE01 is very important to this red cell turnover as it controls sort of hemophagocytosis, that sort of macrophages breaking down these red blood cells and the iron that is subsequently produced. And they were focused on this particular gene because it's known to have a Mendelian form that includes iron overload. But there's also a gain of function mutation in this gene that is pretty common. And this gain of function mutation they found in blood bank participants of African ancestry to have an increased level of iron, particularly in adults. So those over 40, you see here with transferrin and ferritin. Interestingly, the very same variant had recently been associated with protection from malaria, at least in the heterozygous state. And of course, this is a variant that varies from 9 to 18% in frequencies in groups that have been sampled across Africa, but it's actually very rare and not seen very much outside of Africa. And so, here's a variant where you've got this sort of idea of pleiotropism, where, because of the context in Africa, and then you go outside of Africa, there's a new consequence that was relevant, and you needed to have the right people in order to identify this variation. So that's example number one. Example number two, some work we did looking at transfusion alloimmunization in sickle cell disease. So sickle cell disease is a disease of blood. Transfusion of red blood cells is a very important part of the management. And it turns out that some subset of individuals with sickle cell disease develop what are called alloantibodies after each transfusion. So these are antibodies that you develop to some of the minor antigens. So not the big ABO or rhesus, but some of the minor antigens. The vast majority of people don't develop any antibodies, but some do. And that creates a problem because if each time you're given a transfusion, you develop new antibodies, then it becomes really difficult to find compatible blood after a while. So we had the opportunity to perform a genome-wide association study of this sort of responder status. And in that, we were sort of very mindful of what Marylyn just spoke about, which is that there is this continental admixture among individuals of African ancestry. And again, here for these studies, we're working in individuals from the Gulf Coast, so Louisiana, Texas, Oklahoma. And we were aware that most African Americans have about 20% European ancestry as part of their admixture. And so what we wanted to do is to leverage the local ancestry components that Marylyn just spoke about. And just to sort of reiterate that, so here we're looking at local ancestry, and this is sort of the proportion of African ancestry that you can see as we move along chromosome 11. And so all these individuals have sickle cell disease, so you can see that over that sickle cell locus, over the beta globin gene, they're almost 100% African ancestry, which is consistent with the origins of the allele in Africa. But then over the rest of the genome, it kind of bounces up and down to have an average of about 20%. And so what we did was in our association studies, we adjusted for each SNP as to whether or not it was coming from a European haplotype or an African haplotype in the cases and the controls. And when we did that, we found a locus on chromosome 5 that was associated with African ancestry restricted analysis, but not seen with the European ancestry analysis. This was also our top hit in the sort of standard GWAS, and we had replicated it as well. And actually, when we looked at the frequencies, it varies across the African continent, but to this point, we have not seen that variant outside of Africa as yet. So the variant itself sits on the top variant, sits on a ENCODE enhancer with two nearby genes that we're not 100% sure about, but there is this sort of interaction with that we're able to show with an upstream gene called IL-12B. And if IL-12B sounds familiar, it's because it's known to be important in the immune response to intracellular pathogens. And more specifically, this is the same locus that Scott Williams' group had described as being showing resistance to tuberculosis in individuals from Uganda and Tanzania. So again, you've got this idea of potential pleiotropy where a situation in one context has a different context otherwise, but again, would not be seen unless you're looking in the right population group. So I think I'm going to stop there. I just wanted to sort of say that, hopefully we've gotten from this and from all the other talks, the importance of sort of breadth of variation, how much there is, and how much those things create challenges, but also opportunities for medical genomics, precision medicine, and understanding population level disease. So I'm just gonna
go ahead.
Sorry, I just wanted to make sure that I acknowledged the people who did the hard work for H3 Africa. So I am merely the mouthpiece on this side of the Atlantic, but most of the work was done from those in Africa. And then I'm happy to take questions.
Thank you so much, Neil. That was an excellent talk, and you're right, acknowledgements are super important. We have a few questions here. So the first one is, Kelly wanted to know if you could talk more about the research participant recruitment process, and what are the challenges and main reasons of resistance, if there is resistance from participants?
Yeah, so I mean, I guess I can speak to the projects that we've been involved with there, which are in Botswana and in Uganda, but it's very similar across the continent. And in general, there's a very different attitude sometimes to what you get on this side of the Atlantic, which is that people really want to be involved. There's this idea of community and trying to be a good citizen and contributing. So in general, people are the uptake rates for recruitment are actually very high. I think one of the challenges was sort of how to introduce this concept of genetics and DNA and genomics and that kind of stuff. And so a lot of time was spent both, creating advisory boards with the community to sort of partner with them. We had to create, comic books to try and explain the concept of DNA and genetics, particularly to the adolescents that were involved in the study. But then also H3 Africa brought all of the heads of the ethics committees to meetings so that they could have this discussion about how best to get everybody on the same page and to assuage the various fears that there were. So it's a very involved process that occurred on multiple levels.
Okay, that's great. I can understand how complex that is. I have two questions that are related, so we're going to answer this together. One is why there was no representative population from North Africa in the African Genome Project. And then the second person says samples from Western and Southern Africa seem overrepresented in H3 Africa. Does this reflect the human diversity within Africa?
Yeah, so those are great questions. And they're sort of, as you said, very well connected. So again, this was a good example of sort of convenience sampling to begin with. We sort of just said to PIs, if you have appropriately consented samples, please send them in. And we took what we could get. And so just from that, we were able to see a lot of this diversity. But we recognize that we haven't touched very large swaths of the continent. And so in the second phase that we're currently working on now, there's a lot more focus on North Africa, even on sort of Indian Ocean islands as well, but also filling in some of the gaps in Central Africa. So it doesn't cover the diversity. And I'm sure many people will have seen the recent call to have a sort of 3 million genome from Africa to help facilitate the tremendous breadth of diversity. So you're right, it's not there yet.
Well, that's Amboise Wonkum, who's going to be our speaker tomorrow, actually. So we are out of time for now. So we have a break, and if everybody could come back at 25 after sharply, that would be great. Thanks again so much, Neil.
Thank you.